Hi, my name is Dexter Binner, and this is my sliding filament theory. The sliding filament theory was a theory composed in 1954 taking a look at the positioning of myosin and actin at various stages of muscle contraction. Who was responsible for this research? The following four scientists: Andrew Huxley, Rolf Niedergerk, Hugh Huxley, and Gene Hansen. First, we'll be looking at a sarcomere. A sarcomere is known as a basic contractile unit of a myocyte. In other words, it's the part of the muscle fiber that moves. A sarcomere is surrounded by a network of tubules known as sarcoplasmic reticulum, where calcium ions are stored. The sarcomere can be divided into 5 different units. The 5 different units consist of the I band, which contains only thin filaments, the A band, which is found at the center of the sarcomere, which contains both thick and thin filaments, the M line, which contains myomesin, which is a protein, which is found in the middle of the sarcomere, the Z line, which margins the entire sarcomere, where it creates the end zones, and the H zone, which contains only thick filament. Next, we'll be looking at myosin and actin a little bit in depth. Myosin and actin tend to work hand in hand with each other. Myosin is the head and actin is the body. Myosin tends to grab onto actin, resulting in a 4-to-1 ratio. When binding, myosin has an attachment piece for actin, and actin has a binding site for attachment piece. Actin has 2 additional proteins: tropomyosin that acts as a protection by covering the binding site on actin, and troponin, which is a binding site but for calcium rather than myosin. We'll look into calcium ions soon. Now we'll take a look at the composition of muscles. The entire muscle is surrounded by the epithelium, a connective tissue. So beneath the epithelium, a connective tissue, is the permesium, which is another connective tissue, which holds together bundles of muscle fibers known as fascillae. Maybe 10 to 100 muscle fibers per bundle of fascillae. Each bundle is also covered by another layer of connective tissue called endomysium. Endomysium is responsible for covering the sarcolemma, which is a cell membrane of muscle fibers. Myofibrils are singular cylindrical organelles that contain proteins such as actin and myosin. Surrounding the myofibrils are sarcoplasmic reticulum, where the calcium is stored. Each myofibril is built up of repeating subunits called sarcomeres, which we spoke about earlier. Earlier we spoke about calcium and its importance. We'll take a deeper look into that now. Calcium tends to be released from the sarcoplasmic reticulum and binds to the troponin found within the actin. Once the calcium binds to the troponin, the myosin is able to attach to the actin, which pulls the actin, resulting in a muscle contraction. Muscle relaxation is achieved when the calcium ions are removed from the cytosol by an ATP-dependent pump found in the sarcoplasmic reticulum. Without calcium, muscle contraction would not occur. Now we'll take a look at adenosine triphosphate, also known as ATP. ATP's job is to bind to the myosin, and once it binds to the myosin itself detaches from the actin. Once the two detach from each other, the myosin readjusts its position and reconnects with the actin. Reattaching pulls the actin towards the center of the sarcomere, resulting in a muscle contraction. A lack of ATP can result in a condition known as rigor mortis, where the myosin is unable to detach from the actin, resulting in constant contraction. ATP tends to run out quickly during muscle contraction, but it can be produced in 3 ways: a breakdown of glucose, a breakdown of fatty acids, and creatine phosphate found within muscle cells. Now, moving on to the actual sliding filament theory itself. The sliding filament theory can be broken up into 4 stages: the resting stage, the excitation stage, the contraction stage, and the relaxation stage. First, we'll be starting off the resting stage. During the resting stage, The myosin heads are at low energy and an unattached position. The thin actin filaments are also in a relaxed state, and the binding sites for myosin are blocked by regulatory proteins. Excitation stage. When a signal in the form of an action potential from the nervous system reaches the muscle, it triggers a release of calcium ions which bind to the regulatory proteins and expose the myosin binding sites on the actin filaments. Contraction stage. Once the myosin heads have attached to the actin filaments, they swivel towards the center of the sarcomere, pulling the actin filaments towards the center as well. This causes the sarcomere to shorten and the muscle to contract. During this stage, ATP is broken down to release energy for the movement of the myosin heads. Relaxation. When the action potential ceases and the calcium ions are pumped back into the sarcoplasmic reticulum, the regulatory proteins block the myosin binding sites. Once again, the myosin heads detach from the actin filaments and the muscle returns to its original length and relaxes. This is the sliding filament theory.